Ayubowan. Today we are studying the grade 10 lesson, grade 10 science lesson, the first part of the first lesson, that is the chemical basis of life. Here all organisms are composed of chemical compounds. These chemical compounds are made by bonding of naturally existing elements. Normally there are 92 natural elements in Earth, right? Out of them, only 25 elements are present in the living organism. There are four major types of elements as carbon, hydrogen, oxygen and the nitrogen. These chemical compounds can be categorized into two groups as organic compounds and inorganic compounds. Organic compounds mean the compounds which contain carbon are called organic compounds. Compounds which do not contain carbon are called as inorganic compounds. Some examples for inorganic compounds are carbohydrates, proteins, lipids and nucleic acids. And some examples for inorganic compounds are water, mineral and gases. And there is an exception. Some inorganic compounds are there which contain carbon, such as carbon dioxide, bicarbonates, etc. Then we'll separately discuss about the first example under the organic compounds, that is carbohydrates. So if we get carbohydrates, they are the most common or abundant organic compound on Earth. The major elemental composition is carbon, hydrogen, nitrogen and oxygen. In there, carbohydrates are found in potato, sweet potato, grains and sugars, right? So then there's a ratio. The hydrogen oxygen is bind with a ratio of 2:1, and it has a molecular formula of CxH2O. Carbohydrates can be classified into three groups based on the way they are formed. They are monosaccharides, disaccharides and polysaccharide. Monosaccharide means simple sugars. The major properties are they are crystalline shape, water soluble and sweet taste is there, right? Under that the examples are glucose, fructose and galactose. If you get glucose is found in ripened fruits, bee honey, right? And glucose is produced during the photosynthesis of plants, and as a glucose is produced as the end product of starchy food and it is absorbed into the bloodstream. Then the next example for monosaccharide is the fructose, where you can see them in ripened fruits and bee honey, pumpkin, carrots. There we call this fructose as the sweeter sugar and as well as fruit sugar. The third example is galactose, which can be seen in dairy products such as milk, cheese, butter. In these dairy products, galactose it doesn't have a taste. Then the second type is the disaccharide, where two monosaccharides are bind together to form a disaccharide. Here major same properties are there. They are water soluble, crystalline shape and sweet taste. Their examples for disaccharides are maltose, sucrose and lactose. Maltose is found in the germinating seeds. It is formed by union of two glucose molecules. second example is sucrose. Union of glucose and fructose form sucrose, and union of glucose plus galactose form the lactose, right? So in lactose, in this one, it is more sweet taste in sucrose, and if we get the lactose percentage in cow milk, it is like 4% to 6, and in the human milk, it's like 6% to 7%. Then the third group under carbohydrate is a polysaccharide. It is a polymerization of large number of monosaccharides. Here the some properties are they are insoluble in water, and no, it's not in the crystalline shape. Then examples such as are like cellulose. It is found in the plant cell and fibers. Here the cellulose is not digestive in the digestive system, so this is helpful to avoid the constipation. Constipation means it's a disease which that means it has the difficulty to remove the fecal matter. Then the next example is starch, which is the type of carbohydrate that store food in plants. Then the third group is glycogen, where it can be found in animal liver and muscle. It is a form of carbohydrate that's stored in the human body. The test to identify carbohydrate, first test is starch test. So during the starch test, we take grinded starch sample and we add few drops of iodine. Iodine is a brown color solution. So you can see a color change. This turns into a purplish blue color. So this experiment confirms that this food contains starch. Second test is for the glucose. So here we take a glucose sample into a test tube and we add few drops of Benedict solution, which is a blue color solution. Then we mix it well and we immerse the test tube on a water beaker and heat it through the Bunsen burner, right? So you can see the observation. observation is like the color changes from blue to green, then from green to yellow, then gradually color changes to orange. Finally, you can get a brick red precipitate, right? The test for sucrose, here we take a sample of sucrose solution and add it into a test tube. Then we add few drops of phenolic solution, and as earlier we immerse the test tube on a water beaker and heat it using the Bunsen burner and see whether you can see a color change or not. So in this instance you cannot see a color change. So what we do is we add few drops of acid, dilute HCl or sulfuric acid. Then we boil it. So when this boiling step occurs, what happens is the sucrose molecule breaks into glucose plus fructose. Then we add few drops of benedict. After that you can see the observation as a brick red precipitate. The significance of carbohydrates are like first one is as an energy source. Main source to obtain energy is the carbohydrates. Second, as a storage component, where examples like glycogen and starch. Then as a structural component, example cellulose, and as a constituent of nucleic acid. Our next organic compound is the protein. So proteins contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. This is a complex molecule which is made with polymerization of large number of amino acids. The foods which are rich with protein are such as meat, fish, egg, right? So if you get the significance of protein as an energy source, when lipid and carbohydrate energy sources are not sufficient, protein is used to generate energy. Then to make the structural component, like hair, feathers are formed with the composed of the keratin protein, right? And as enzymes, biochemical reactions take place in organisms are catalyzed by the enzymes, right? So these enzymes are also proteins. As hormones, some hormones are proteins. Then as antibodies, these antibodies are produced to protect the body against harmful microorganisms that enter into body, right? Then the test to identify starch. So next is the test to identify protein. Here we take grinded dal or a egg yolk, add it into a test tube, and we add sodium hydroxide solution and copper sulfate. Then we mix the solution well, right? Then you can see an observation that a purple color will develop slowly. So from this experiment, it confirms that if this observation is there, it confirms that this food contains proteins. Next, we have a simple topic called enzymes, right? These are special proteins that are produced within all organisms that is used to increase the biochemical reactions. So these are called enzymes, right? First, we'll see the activity of amylase on starch. So an activity, how amylase act on starch, right? So here we take starch solution, right, and we add amylase solution. This amylase is extracted from the germinating green gram seeds, right? Then we take a drop of this solution onto a porcelain tile, right, and we add a drop of iodine solution to this, right, and see the color change. We do this in 2-minute intervals for 20 minutes, right? So observation is, right, the blue color obtained from this mixture gradually reduces, right, with time, and finally obtain the color of the iodine. Starch gives a black color with iodine, but there's no color change after 20 minutes. That is because all the starch which is present in here has been converted to maltose by the amylase enzyme here. Right? We will stop the lesson now. So in the next day we'll study what are lipids and what are the test for lipids and the nucleic acids. Thank you.